Thank you very much. Oh, you had one more. You want to slide with your collaborators?
Yeah.
So we can show. Okay. So I hope that the connections are clear and we're seeing already the benefits. We hope that the patients will see the benefits because cancer has touched all of our lives, personally, professionally, and at the end of the day, the key point is the patient is waiting. The patient is waiting for a cancer therapy that isn't mutilating the body through surgery, isn't poisoning the body through chemotherapy or radiating the body with even the most modern radiation techniques that still kill peripheral fast-growing cells. And that is what the promise of bacterial peptides bring for the patient. ICGB is bringing benefits to patients all around the world by bringing training both locally in developing countries like South Africa, India, and Thailand, and also in Trieste, where young promising scientists like Jenny Safini, who, Marco, I hope during Q&A someone will ask, as I understand she is a brilliant young scientist from Morocco who now will have the skills for IP management, technology transfer, and will be heading her own lab when she goes back to Morocco after her PhD. We'd like to open it up for questions now, and hope also there's an opportunity for personal interaction and to stay in touch on these very important issues. Thank you. Oh, and they've asked if you can come up to the microphone if you have a question and introduce yourself.
Gary Keller. And so first I just wanted to thank you. I didn't catch the whole panel, but what I did catch was very interesting, and it's always wonderful to hear Dr. Charpard talk. But specifically in reference to what you put forward about cancer therapeutics and finding more holistic ways that are less invasive and have better efficacy, I appreciate that as someone who has founded a cancer therapeutics company based on natural killer cells. And so if I look at it from that perspective, if we look at the first therapy for cell therapy, aside from bone marrow transplant, which was really the first cell therapy that was approved, lymphocyte-activated killer cells were exactly done because they were stimulated to make the body respond. And I was wondering if you see that it's really the same type of mechanism that you're experiencing through these biofilms or the use of some of the various bacteria that you're using in your research.
I mean, you raised very important points. You know, in general, as you rightly said, that people have been trying not only to develop drugs, but also to bolster the immune system. Note that bacterial proteins, as I said, they're intelligently designed by the bacteria not to harm the host, but just to keep in check the invaders like cancer. So they have structural similarity with immunoglobulins. So they are not recognized, even though they are foreign proteins in the body, they are not recognized by the immune system. So they have no, as far as we know, now we haven't conducted studies regarding exactly how these proteins work. All we have done is we have looked at, so they have two, we could go into details, they have two modes of action. One is they can go inside, inside the cell, and once inside the cell, they can form complexes with tumor suppressor P53 to induce apoptosis. They can form complexes with the receptors of vascular endothelial growth factor, prevent angiogenesis, and things like this. But there is a pumping mechanism. Cancer cells are very smart. They can pump out anything that goes in. So the bacterial protein doesn't just depend on going inside. They actually form complexes with what's called receptor tyrosine kinases that are hyper-expressed in many cancer cells. So they don't have to go in. And they can bind those with a KD value that's better than the ligand binding, and will prevent activation of the receptor signaling systems. So you are asking do they also work by activating any kind of immune mechanism? I do not know. We haven't really looked at all this. But the net effect is that I mentioned to you that there were 2 patients at higher dose of the peptide. Tumors, metastatic refractory tumors that are not responding to any conventional drugs were wiped out.
The other thing I just was going to say, because as I listen to this, which is, this is, I don't think it's an unusual phenomenon. I think it's a great thing to figure out how we could actually look and apply it in a clinical setting. As I look at that, there seems to be some challenges. One is the regulatory pathway is pretty austere in regard to being able to get approval. And the other that occurs to me from issue would be, like you said, they could pump it back out. And if you're doing bacteria, are you going to induce sepsis? Because if you do that, the problem is we don't have any good therapies. So if we go from cancer to sepsis, then I think we've gone from bad to worse. And it's just, and I don't know that would or wouldn't happen. It's just a consideration.
Yeah. So right now, I'm in touch with a friend of mine in the Burma. He's in Salk Institute. He has a mouse model where he can actually activate some of the mutant genes that would contribute to the formation of glioblastoma multiforme, the brain tumor I mentioned to you. So I said, in that, I have this bacterial protein, seems to be non-toxic, seems to prevent cancer initiation and kill any preformed tumor. So if I express it in the mouse genome, in the glial cells, what the tumor, glioblastoma, occurs, if I express them in the mouse glial cells, will it prevent glioblastoma? And now we are talking about expressing bacterial proteins initially in mice and perhaps later in human. If my genome will allow expression of a bacterial protein that will keep cancer in check, I don't need any therapy. That protein produced in my body will keep the cancer in check. This is a long-term goal, no doubt about that. But if the mouse experiment with glioblastoma, by expressing these bacterial proteins in the glial cells of mouse will prevent the onset of glioblastoma, that's a very good first step for me to go and think about doing it at some point in time. So there are various approaches, you know. I mean,
And also, if we could just add with the stent.
no, that's a device. The other thing
It
is,
stops the bacteria from entering the body that would prevent sepsis.
so there is no medical device for treating cancer. So if we can, in addition to the drugs, if we can put a stent with the bacteria encased in a membrane that wouldn't allow the bacteria to go to the bloodstream, but would allow the protein wafers to be secreted because the bacteria feels the presence of the tumor and would keep on sending the proteins. So we are talking about multiple ways to treat cancer, but the ideal way would be to express some of those bacterial proteins in our own genome that will keep the cancer in check.